Since the early studies of Aristotle, -300 BC, we have been fascinated by chicken embryogenesis. A small window cut in the eggshell allows observation of the embryo during its development. The chicken, Gallus gal/us, is a vertebrate that undergoes morphogenesis and organogenesis, growing and developing remarkably similar to humans. Accessibility of chick embryos to experimental manipulation not possible in mammalian embryos has made the chick a powerful teaching and research tool. Thus, the chick is an excellent model to understand human birth defects. In addition to these attributes, chicken eggs are available worldwide, year-round in large numbers and at low cost. Because the chicken is such a favorite model system, much is known about its morphology, teratology, and experimental embryology. The chicken is also an agriculturally important organism. The poultry industry in the United States is a multi-billion-dollar industry. Another avian model system is the Japanese quail, Coturnix japonica. It develops similar to the chicken. Quail embryonic cells can be grafted onto a developing chick embryo to create a chimeric embryo called a chick-quail chimera. The quail cells survive and integrate into the chick's tissues as they develop. Quail cells are readily discerned from those of the host chick, since quail cell nuclei have a distinctive nucleolus and quail cells express cell-specific antigens not found in the chick. The quail cells serve as genetic markers in cell lineage studies that determine cell fates. Much information has been gained about basic development from chick-quail chimeras. Now that sequencing of the human genome is completed, researchers are focusing on understanding individual gene function, and how groups of genes function in concert. It has been estimated that about half of the known predicted human genes encode proteins of unknown functions. The focus for researchers today is to answer questions like, where and when is a gene expressed in the embryo? This can be answered by in situ hybridization, a technique that visualizes a specific gene or mRNA in an intact embryo or tissue, and a technique called immunofluorescence microscopy that localizes specific proteins in cells, tissues, or intact embryos. In order to interpret the data, an understanding of basic morphology is needed. Other questions include, what is the function of a gene? With which other genes does it interact? These can be addressed using gene misexpression studies, microarray analysis with chicken gene chips, profiling, and transgenic chickens. The chicken has also had its genome sequenced recently. Thus the chick embryo is one of the best models to answer these questions because embryos can be manipulated in vivo, the egg is naturally sterile, and embryos are amenable to genome analysis. Studying chick embryology is a window to understanding mammalian development. Comparative genome analyses can be used to analyze diseases of birds, such as severe acute respiratory syndrome, SARS, that relate to humans. Unlike frog embryos, chick embryos are relatively transparent at early stages of development. This has the advantage of observing the spatial relations of different structures in various planes throughout the embryo. Since all structure residential are seen simultaneously in intact embryos, whether in focus or not, focusing up and down on the embryo helps immensely to discern structures. As described in Chapter 2, embryos are typically studied as whole mounts or sections. Whole mounts are thick and can be easily crushed during viewing. Slides of serial sections are delicate and care must be taken not to exert any pressure on the cover slip as this can shatter sections even though the slide appears unharmed at first glance. Damage to one slide in a serial section series ruins the entire series. A reliable approach to studying chicken development is to first understand the orientation of structures in whole mounts before studying corresponding serial sections. An excellent strategy is to trace structures in sections for one organ system while temporarily ignoring others, and then going back to trace components of a different organ system. For the beginning student, it is hard to understand the implications of the fact that no two embryos are exactly alike. Due to variability as to when eggs are laid, embryonic development can differ slightly. In addition, Not all regions of the avian embryo develop at the same rate. Cranial structures are initially more developed than caudal structures. Thus it is useful to examine multiple embryos to gain full understanding of chick development. Although great care is taken to orient embryos the same way during sectioning, the angle at which section planes are cut is greatly variable. This must be considered when naturally curved embryos are studied. Analysis of slides of a serially sectioned embryo indicates that all of the structures are there, but often in a slightly different spatial arrangement than that of a different serial section series. The previous chapters have followed production of chicken gametes and the development of the fertilized chicken oocyte to the Neurula stage. Chapter 1.1 assumes familiarity with the terms in these chapters. A review of the 24-hour chick embryo, Chapter 8, will be useful before proceeding with the stages shown here in Chapter 1.1. Specific stages of chick development were chosen to demonstrate features of the organ systems. Early stages are identified in figure legends according to the embryo's number of somates, when possible, and age, in addition to the Hamburger and Hamilton staging series, Table A.9. Although the embryos of Chapter 11 represent selected snapshots in time of chicken development, it is helpful to remember that the embryos are continuously rapidly developing. Typical early stages are shown in detail, with selected later stages. External morphology. Overview. It takes 21 days for a chicken to hatch from the egg. During this time the embryo undergoes extensive changes in which differentiation, morphogenesis, and organogenesis form the embryo's tissues and organs. By 16 hours of development, the gastrula appears as a small streak with its longitudinal axis perpendicular to the long axis of the egg, Chapter 7. At 24 hours, the flat, linear embryo is stretched over the yolk with its ventral side facing the yolk, Chapter 8. The embryo remains linear in shape up to about 38 hours. Beginning at Rees-Rhyme, several processes including torsion and flexion dramatically change the embryo's appearance, and its position with regards to the yolk. Torsion is the twisting of the body about its long axis, such as when a golfer takes a large swing at the ball. Torsion occurs slowly and starts with the rostral area of the embryo and progresses caudally. It is complete by 4 days of development so that the embryo's entire left side lies against the yolk. Fcation refers to a bending of the body about a transverse axis, such as when one bends the head at the neck toward the chest. Flexion causes the embryo to appear C-shaped with its head and tail lying close together at 4 days of development. The embryo undergoes flexion in 5 regions. Cranial flexure, a bend at the mesencephalon so that the forebrain prosencephalon bends ventrally towards the yolk, begins at 38 hours, and by 72 hours is at 90 degrees. Cervical flexure, a ventral bend at the transition between myelencephalon and spinal cord, future neck, begins at 48 hours and by 72 hours is at 90 degrees. Pontine flexure, a ventral bend in the mid-region of the rhombencephalon, between the metencephalon and myelencephalon, that is visible about 5 days. Dorsal flexure, a ventral bend in the mid-body region that makes the back convex dorsally, begins at 72 hours. Caudal flexure, a ventral bend in the tail so that the tail curls on itself, visible at 72 hours. Body folds. 3 body folds form which establish the contour of the embryo and separate the embryo from the yolk. 1. Head fold, cephalic fold, 2. Lateral body folds, and 3. Tail fold, caudal fold. The body folds help separate the intraembryonic layers, body tissues, and extraembryonic layers, those that do not contribute to the body. The head fold, which shapes the head forms first and develops AR 24 hours, stage 6, as seen in Chapter 8. The space underneath the head fold is the subcephalic pocket, Figs 8.25 and 8.35. The 2 lateral body folds develop next and define the embryo's sides. The tail fold forms last and separates the tail from the yolk by 43 hours, Fig 11.17. Extraembryonic membranes. Recall from chapters 6 to 8 that 3 regions can be discerned in the early chick embryo: 1. Area pellucida, a lighter area that contains 2. The embryonic axis, and 3. The area opaca, which appears dark due to underlying yolk. The area opaca surrounds the area pellucida. Beginning at 24 hours, the area opaca closest to the area pellucida becomes mottled with dark spots and streaks. These are mesoderm cell aggregates that form blood islands, which are the beginnings of primitive blood cells and blood vessels. The blood vessels originating in the yolk sac, extraembryonic membrane enveloping the yolk, are the Viklin blood vessel, which are important in acquiring nutrition from the yolk. The part of the area opaca containing blood islands is called the area vasculosa. At first the region outside the area vasculosa lacks blood islands and blood vessels, and is called the area vitellina, Figs 11.2, 11.7, 11.10, 11.16. A circular blood vessel called the sinu terminalis separates these two regions of the area opaca, Fig 11.2. The area vitellina will surround the yolk by 72 hours. The embryo develops 4 sets of extraembryonic membranes, Fig 11.1. Yolk sac first extraembryonic membrane to appear. Functions to house the yolk, and spreads over the yolk to form a sac open to the embryo's gut. Forms from the area opaca and develops from splanchnopleure. It is continuous with the intraembryonic splanchnopleure that forms the walls of the gut. Yolk forms the gut floor. Amnion, thin extraembryonic membrane made of somatopleure. First visible at 30 hours in the head region as a fold of somatopleure that eventually completely surrounds the embryo in a fluid-filled sac by 4 to 5 days. Functions in a protective role, keeping the embryo moist. Allantois, a vesicle that begins to form by 72 hours as a small diverticulum of splanchnopleure are from the wall of the hindgut. The diverticulum expands to a large vesicle that eventually encompasses the embryo and yolk sac by 9 days. Functions in gas exchange and as a reservoir for wastes such as urea and uric acid. Chorion, a thin extraembryonic membrane that is derived from somatopleure. It is the outermost membrane and surrounds the entire embryo, amnion, and allantois. It lies against the eggshell and functions in respiratory exchange of gases. In addition, the chorion transports calcium ions from the eggshell to the embryo that are needed for beak and skeletal formation. When the allantois lies up against the chorion, this combined membrane is referred to as the chorioallantoic membrane. Highlights of other MAJ or changes that occur in the avian embryo's external appearance are included here briefly. 1. The embryo lengthens and enlarges at first, easily recognized by an increase in the number of somates. The heart appears as a slight bulge that bends to the right at 33 hours and later enlarges and twists on itself. Eyes appear, the rudiments of which first begin to form at -28 hours and occupy a large area of the head in later stages. 4. Ears form. 5. Limbs develop from limb buds by 72 hours. The wing buds appear lateral to somates 15 to 20, and the leg buds appear later at the level of somates 27 to 32. 6. The face forms. Beginning at day 4, the maxillary and mandibular processes of the first pharyngeal arches grow and fuse in the midline to form the upper and lower jaws, respectively. 7. The beak forms. 8. The embryo becomes less transparent as tissues and organs increase in size. 9. The epidermis becomes opaque as it thickens, and develops feathers on the body and wings, and scales on the legs and feet. Internal morphology. Nervous system. The neural tube forms from the neural plate during neurulation, Chapter 8. At 28 hours, the neural tube differentiates into the future spinal cord, caudally, and 3 brain regions, cranially prosencephalon, forebrain, mesencephalon, midbrain, and rhombencephalon, hindbrain, Figs 9.2, 11.2 to 113. In transverse sections of 28-hour and 33-hour chicks. The prosencephalon is the first brain region encountered as one traces the sections caudally from the first section, Figs 11.4, 11.15. Each brain region has a corresponding cavity, the prococele, mesocele, and rhombocoele, respectively. These are visible in 28 to 38-hour embryos. As they expand, the three brain regions develop swellings and constrictions that divide the brain into 5 regions by 48 hours, Figs 11.19, 11.20, 11.22, 11.23, 11.25. Telencephalon. This is the rostralmost brain region, and is the first region of the prosencephalon. The cavity of the telencephalon is called the telecele. By 72 hours, the walls of the telencephalon evaginate rostrally and laterally, enlarging to form 2 lateral cerebral vesicles that will form the future cerebral hemispheres. The 2 cerebral vesicles are continuous with the lamina terminalis, a medial relatively unexpanded portion of the telencephalon. The cavities of the cerebral vesicles form the paired first and second brain ventricles of the adult. They connect to the cavity of the lamina terminalis, original telecele, by the foramina of Monro. The cavity of the lamina terminalis forms part of the third brain ventricle. The telencephalon also gives rise to the olfactory lobes, which process information concerning the sense of smell. To find the telencephalon in transverse slide sections, first study the corresponding hole mount for orientation. View the transverse sections of 48 to 72-hour embryos tracing the brain from the first section in the series, Figs 11.25, 11.38. Recall that the first brain region in these older embryos is a region of the mesencephalon or rhombencephalon due to flexion. Although tracing sections deeper into the embryo, i.e., caudally, the cranial area of the brain will be traced rostrally due to flexion. The brain will dilate laterally and the rudiments of the eyes will be visible as one moves deeper into the slide series. The telencephalon will be visible just before the head disappears. The cerebral vesicles can be located using this method by first locating the eyes in 96-hour transverse sections and tracing the brain rostrally from there, Fig 1.45p. Diencephalon. This is the second rostral brain region, and is the second, caudal, region of the prosencephalon. It is readily distinguished by the optic vesicles, future eyes, and appears laterally compressed in transverse sections. The walls of the diencephalon thicken to become the adult thalamus and hypothalamus. The roof of the diencephalon forms part of the anterior choroid plexus that secretes some of the cerebrospinal fluid that fills the brain ventricles to bathe the brain and spinal cord. By 48 hours, the diencephalon develops a ventral evagination called the infundibulum, which forms the posterior pituitary. Neurohypophysis. Rathke's pouch, an ectodermal invagination from the roof of the mouth gives rise to the anterior pituitary, also known as the adenohypophysis, Figs 11.25 EF, 11.36b, 11.38, 11.43c, 11.45 L. A small mid-dorsal evagination called the epiphysis appears from the roof of the diencephalon at 72 hours, and this becomes the adult pineal gland, Figs 11.29 to 11.33, 11.38p. The cavity of the diencephalon, which is called the diacocolc, forms part of the third brain ventricle. Mesencephalon. This is the third brain region, midbrain, and it is located between the prosencephalon, diencephalon, and rhombencephalon, metencephalon. The mesencephalon greatly expands by 72 hours, stage 18, and is distinguished from the rhombencephalon by the isthmus, a narrow brain region. The roof, dorsal side, thickens to become the adult corpora quadrigemina that are involved in visual, optic lobes, and auditory reflexes and processing. The floor of the mesencephalon thickens to become the crura cerebri that contain fiber tracts that connect the forebrain to the hindbrain and spinal cord. Due to thickening of the walls of the mesencephalon, the large mxococolc cavity of the mesencephalon becomes a narrow opening called the cerebral aqueduct, aqueduct of Sylvius, in the adult. In transverse sections of 48-hour embryos, the mesencephalon may be the first brain region encountered, and appears distinctly round in transverse sections of later stage embryos, Figs 11.38, 11.45, 11.47. Metencephalon. This is the fourth brain region, and is the rostral part of the rhombencephalon. It is located between the mesencephalon and myelencephalon. Its rostral boundary is the isthmus, and its caudal boundary is not well defined for the first 4 days. In whole mounts and transverse sections of 48 hours or more, the thicker roof of the metencephalon helps distinguish it from the myelencephalon. The cavity of the metencephalon is the metacoccale, which becomes part of the fourth brain ventricle. At 4 days the metencephalon appears relatively unchanged, but after that it grows dorsally to form the cerebellum and ventrolaterally to form the pons and cerebellar peduncles. Myelencephalon. This is the fifth brain region, and is the caudal area of the rhombencephalon. It is located between the metencephalon and spinal cord. There is no distinct boundary between the myelencephalon and spinal cord for the first few days of development. The myelencephalon is characterized by several paired neuromers, which are small segments formed by constrictions of the wall of the myelencephalon, Figs 11.36, 11.37. The sixth neuromers are long. Although transient, neuromers are involved in patterning of the brain. The cavity of the myelencephalon is the myclm, which becomes part of the fourth brain ventricle. The thin dorsal roof and thick walls and floor of the myelencephalon make it easy to identify in whole mounts and sections of 48-hour or older embryos, Fig. 11.25 dm. The roof obtains a rich blood supply that forms the posterior choroid plexus that contributes to production of cerebrospinal fluid. The lateral and ventral walls develop into the medulla oblongata that serves as a reflex center for involuntary activities, i.e., breathing, and a main pathway for connections between the brain and spinal cord. Due to increased flexion of the embryo, the very first transverse sections of 72-, 80-, and 96-hour embryos usually pass through the myelencephalon, Figs. 11.38, 11.45. The full extent of the large myelencephalon can be seen in whole mounts and sagittal sections of 72-, 80-, and 96-hour embryos, Figs 11.29 to 11.32, 11.35. 11.36, 11.39, 11.40, 11.42, 11.43. Spinal cord. The remainder of the caudal neural root develops into the spinal cord. In transverse sections at 48 hours, the spinal cord appears elliptical and slightly compressed laterally, Fig 11.25 NV. Its lumen, cavity, is derived from the neurocoel, like the brain ventricles, and is called the pineal canal, central canal. The spinal canal becomes very small and round by 8 days, Fig 11.51c. The lateral walls of the spinal cord become thicker than its dorsal roof plate, and ventral, floor plate, walls. By 72 hours, the spinal cord wall has differentiated into an outer marginal layer characterized by many fibers and few cell nuclei. This will become the future white matter of the spinal cord, Figures 11.51c, 11.53b. The innermost layer of cells adjacent to the spinal canal is the ependymal layer. The layer that develops in between these two layers by 4 days is the mantle layer, which appears white and dark since it is heavily nucleated. The mantle layer becomes the gray matter of the spinal cord, Figs 11.51c, 11.53b. The dorsal half of the spinal cord is the alar plate, and the ventral half forms the basal plate. By 72 hours, several paired spinal ganglia, dorsal root ganglia, appear lateral to the spinal cord. In transverse sections at 72 hours, the spinal ganglia appear as dark spots lateral to the spinal cord, Fig 11.38. By 4 days, spinal nerves begin to develop and connect to the spinal cord by a dorsal root, sensory afferent, and a ventral root, motor efferent. The spinal ganglion is present on the dorsal root of the spinal nerve, before the dorsal root joins the ventral root, Figs 9.5. 11.53b. A thin branch called the ramus communicans extending from the combined dorsal and ventral roots connects with the sympathetic ganglia. The sympathetic ganglia appear as dark spots dorsolateral to the dorsal aorta in transverse sections of 4 and 5 day embryos. Figs 1.45, 1.48. The spinal nerves are difficult to identify in sections of embryos that are 4 days old and younger. Cranial ganglia and nerves as described in Table 9. 1. The chick embryo develops 12 pairs of cranial nerves derived from neural crest and several placodes. A review of Table 9.1 at this time will aid in understanding chick cranial nerve development. The cranial nerves do not develop at the same time, so that some appear earlier and more prominent than others. The earliest forming cranial nerves begin to appear by 72 hours, and most are visible by 96 hours. Cranial ganglia are associated only with cranial nerves V, 7, 8, 9, and X, and appear as a dark mass associated with the hindbrain. The discussion below includes the cranial nerves and ganglia that are visible up to 96 hours, spinal nerves 4, 6, and 11 are too difficult to find or appear at later stages not shown in the atlas. Cranial nerve I. The olfactory nerve is first visible on day 4 in transverse sections as small fibers emanating from the nasal placode, nasal pit, growing toward the region of the telencephalon that will form the olfactory bulbs. Cranial nerve 2. The optic MRVE originates from the retina and relays visual information to the brain. Fibers of the optic nerve can be seen making their way to the optic lobes of the brain as they go straight or cross to the opposite side of the brain via the optic chiasma, also called optic chiasm, Fig 163. This provides the means for depth perception. Cranial nemi 3. The oculomotor nerve appears as two dark streaks emerging from the floor of the mesencephalon in transverse sections of 72-hour and later chick embryos. Figs 1.38, 11.45m, 1.47a. This nerve migrates ventrally to innervate 4 pairs of eye muscles, superior, inferior, and external recrus, and inferior oblique muscles. Cranial nerve V. The most prominent cephalic ganglion that is visible in whole mounts and sections at the level lateral to the ventral region of the rostral myelencephalon is the semilunar ganglion, Gastrulation. Ganglion, of the trigeminal nerve. It is visible in 56-hour whole mounts, Fig 1.26. By 72 hours, it splits to form a cranial branch called the ophthalmic nerve and a caudal branch that will later give rise to a lateral mandibular nerve and a medial maxillary nerve by 4 days. These nerves can be traced to the first pharyngeal arch in transverse sections of 72 and 96-hour embryos, Figs 1.38, 1.45. Cranial nerve 7, VLLL. At the rostral side of the otic vesicle is the acousticofacialis ganglion, a combined mass of ganglia from cranial nerves 7 and 8, Fig 11.23d. On day 4, the ganglia become separate and will develop into the adult acoustic and geniculate ganglia. The facial nerve, 7 will innervate the second pharyngeal arches, and the auditory nerve, 8 will innervate the inner ears. Cranial nemes 9 and X. At the caudal side of the otic vesicle is the very small, round superior ganglion of the glossopharyngeal nerve, Fig 1.23d. This nerve will migrate to the third pharyngeal arches to innervate the pharyngeal muscles and salivary glands of the adult. Lying caudal to the glossopharyngeal nerve is the jugular ganglion of the vagus nerve, X, Fig 1.38f. The vagus nerve has not yet formed at 72 hours, but will innervate the fourth pharyngeal arches. Cranial neme 12. The hypoglossal nerve is first visible in the 4-day chick embryo, Fig 1L.45c. In transverse section, it emerges from the ventral side of the myelencephalon to extend caudally to enter the pharynx where it will innervate the muscles at the base of the tongue. Sensory systems. Olfactory system. The olfactory system arises from paired olfactory placodes, nasal placodes, that are thickened areas of ectoderm that lie to each side of the telencephalon, Figs 11.29-32. They are visible rostral to the eyes in 48-hour transverse sections, Fig 11.25 fg. The olfactory placodes invaginacy to form depressions called olfactory pits, nasal pits, visible in 72 and 96-hour transverse sections, and 96-hour whole mounds, Figs 1.1.38, L 1.40c, 11.45 n. The olfactory pits are continuous with the amniotic cavity and form the external narcs. The olfactory pits deepen and initially open to the oral cavity, but become attached to the caudal region of the mouth as internal nari when the palate forms by day 7. The epithelium lining the olfactory pits becomes the odor-detecting olfactory epithelium that is the source of olfactory nerves. Visual system. The beginnings of the eyes first appear at about 28 hours as the optic vesics, which are lateral swellings of the diencephalon area of the prosencephalon, Fig 1.61. The cavity of the optic vesicle is the opticocle. The optic vesicles constrict at the base to form the optic stalk visible in transverse sections at 33 and 48 hours. When the distal part of the optic vesicle reaches the lens pleuronda, a thickening of the ectoderm that forms in response to inductive signals from the optic vesicle, signals from the lens placode induce the optic vesicle to invaginate to form the optic cup, which becomes double-walled by 48 hours. The walls of the optic cup form layers of the retina. The outer layer is the pigmented retina and the inner layer becomes the light-detecting sensory retina. Later the retina differentiates into several layers. The retina generates the optic nerve. Fibers of the optic nerve enter the optic stalk around day 3, Figs 1.38 L, 11.45 m. During formation of the optic cup, the opticoele is eliminated and a new cavity, the future vitreous body of the adult eye, is formed. Cells of the optic cup secrete vitreous humor. The optic fissure, choroid fissure, is a small transient gap in the ventral wall of the optic cup. It allows a blood vessel to supply the eye, Fig 11.43e. A mesodermal ridge carrying the blood vessel into the eye through the optic fissure enlarges by day 5 to form the pectin, which may be involved with increased oxygenation and nutrition for deep ocular tissues, Fig 11.61g. The optic vesicle/optic cup induces the lens placode to pinch off from the ectoderm to form a lens vesicle, which becomes the transparent lens of the adult eye. The lens vesicle resides in the optic cup and differentiates into lens epithelium and lens fibers that can be seen growing within the bobby of the lens by 72 hours. The ciliary body and iris develop from the lips of the optic cup around day 8. The iris is yellow-orange in adult chickens, Fig 1.18. Contraction of ciliary muscles changes the shape of the lens, allowing it to accommodate visual images. As the lens migrates inward, it loses contact with the epidermis and forms a space that is the anterior chamber of the eye. The epidermis overlying the anterior chamber becomes the transparent cornea of the eye. Mesenchyme surrounding the optic cup differentiates into the outer coats of the eye. The choroid, thin inner layer chat, produces nutrition for ocular tissues, and sclera, outer layer, which is the white of the eye. Eyelids begin to form day 7 from a circular epidermal fold. By day 10, a fold within this fold becomes the nictitating membrane, a thin protective layer that rests against the cornea, Fig 11.611. In summary, the optic cup generates the retina, iris, and ciliary body, the ectoderm produces the lens, cornea, eyelids, and nictitating membrane, and the mesenchyme forms the sclera and choroid. Auditory and vestibular systems. The inner ear develops from the otic placode, auditory placode, that lies to each side of the myelencephalon. The otic placode is visible in 33-hour embryos as a thickened region of ectoderm, Fig. LL.15 HI. By 48 hours, the otic placode has invaginated into the embryo to form the otic pit, auditory pit, which pinches off from the epidermis by 72 hours to form the otic vesicle, auditory vesicle, otocyst, Figs 11251, 11.38 CE. A dorsal evagination of the otic vesicle forms a blind-ending outgrowth called the endolymphatic duct visible at 72 hours, Fig 11.29. Beginning at 4 days, the otic vesicle forms bulges. Dorsally it forms the utriculus that will become the cranial and caudal semicircular canals, and a lateral bulge becomes the horizontal semicircular canal. Fig 1151a. Ventrally the otic vesicle forms the sacculus and cochlea. The middle ear and outer ear develop from the first pharyngeal pouch, tubotympanic recess, and surrounding wall of pharynx. A narrow passageway called the eustachian tube forms between the pharynx and the middle ear. The external auditory meatus, the opening of the eustachian tube to the outside, appears by day 6 and is shown in this atlas at day 9, Fig 11.52. Birds form only one auditory ossicle, ear bone, the columnar ja, stapes, located in the middle ear. A cartilaginous form of this bone appears by day 6. Unlike mammals, birds lack an external pinna, ear lobe. Digestive system. Adult anatomy. Before studying the embryonic gut, it is helpful to understand anatomy of the adult avian digestive system. Because birds lack teeth, their food passes from the mouth, oral cavity, and pharynx directly into the esophagus, where it is temporarily stored in a small pouch called the crop. When needed, food can be regurgitated, such as during feeding nestlings, or it can pass to the next region of the digestive system, that is the cranial end of the stomach, called the proventriculus. The proventriculus is glandular and contains digestive enzymes. Food next passes to the gizzard, which is the caudal muscular portion of the stomach that grinds up the food. Food next enters the duodenum of the small intestine, and then the coiled remainder of the small intestine, ileum, before passing into a short, straight large intestine. Instead of passing into the rectum next as in mammals, the intestinal contents next pass into the cloaca before leaving the body through the vent, anus. The cloaca is a chamber that is shared with the urogenital system, thus, the cloaca receives fecal material and products of the urogenital system. Overview of embryonic gut. Recall from Chapter 9 that endoderm contributes to the lining of the gut, and the surrounding mesoderm transforms into the muscular walls of the gut. The gut of the early chick embryo, 24 hours, is very simple. Its roof is formed by endoderm and its floor is the yolk. As described in Chapter 9, the body folds help shape the gut, making it appear tubular. By 24 hours, the head fold helps create the foregut, which opens to the yolk caudally via the cranial intestinal portal, Fig 8.25. The foregut is flat dorsoventrally, and wide laterally in transverse sections of 24 to 33 hour embryos, Figs 8.37, 11.4, 11.15. The full extent of the foregut can be seen in sagittal sections of 24 and 33 hour embryos, Figs 8.35, 11.13. The lateral body folds form the midgut region, which is open to the yolk. The tail fold shapes the hindgut, the cranial end of which opens to the yolk via the caudal intestinal portal, Fig 11.25b. By about 48 hours, the embryonic gut consists of a foregut, midgut, and hindgut region, Fig 11.25. The foregut generates the pharynx, esophagus, stomach, and duodenum, essentially all the structures cranial to the liver in the adult. The hindgut gives rise to the large intestine. In the adult this is caudal to the cecum, one or more blind pouches at the junction between the small and large intestines. The midgut generates the intervening portion of the gut, the small intestine and cecum. The endoderm also produces several accessory digestive glands, salivary glands, liver, pancreas, and other glands that do not become part of the digestive system, example thymus, thyroid, parathyroid glands. Individual regions of the digestive system will be described next in the order from cranial to caudal. Oral cavity. A cranial evagination of ectoderm called the stomodium, forms by the second day and meets the endoderm of the cranial foregut, Figs 1.23b, 11.25g. A thin layer of ectoderm and endoderm called the oral plate, stomatodial plate, lies between these areas. The oral plate breaks down by 72 hours so that the stomaderm and foregut are continuous, forming the oral cavity of the mouth, Figs 11.36a, 11.381. The tongue forms after the fourth day from two swellings in the pharyngeal floor. Tuberculum impar, a swelling between the first and second pharyngeal arches that gives rise to the fleshy part of the tongue, and the two, copula, a swelling formed on the lower end of the second and third pharyngeal arches that forms the root of the tongue. Pharynx. This is the most cranial region of the foregut that lies immediately caudal to the mouth. In 48-hour embryos, the pharynx is located ventral to the myelencephalon and somewhat dorsal to the heart, Fig 11.23. The pharynx is surrounded by the aortic arches, to be discussed later. A series of lateral paired evaginations from the pharynx form the pharyngeal pouches, visceral pouches, bronchial pouches, the derivatives of which are described in Table 9.2. The derivatives include the parathyroid glands, alternobronchial glands, ultimabranchial body, and thymus. Recall from Chapter 9 that the pharyngeal pouches meet corresponding ectodermal invaginations, the pharyngeal grooves, visceral grooves, bronchial grooves, Fig 11.24g. Closing plates of fused endoderm and ectoderm are usually present between these structures. If the closing plate is broken, then the resulting opening is called a pharyngeal deft, visceral cleft, bronchial cleft. Between pharyngeal pouches are the pharyngeal arches, visceral arches, bronchial arches, which house nerve, cranial nerve, muscle, blood vessel, aortic arch, and skeletal elements, cartilage/bone derived from neural crest and mesoderm. The pharyngeal arches are mesodermal bands that pass ventral to the pharynx to join with the opposite corresponding pharyngeal arch. Each pharyngeal arch is covered externally by ectoderm and internally by endoderm. The pharyngeal arches and pouches are named in a cranial to caudal direction. Pharyngeal arch 1 is cranial to pharyngeal pouch 1, next comes pharyngeal arch 2 followed by pharyngeal pouch 2, followed by pharyngeal arch 3, etc. The relationship of pharyngeal arches and pouches is seen in transverse sections of 48 to 96-hour embryos, Figs 11.25, 11.38, 11.45. Caudal to the fourth pharyngeal pouches, the foregut narrows ventrally to form the laryngotracheal groove that is the region where the digestive and respiratory systems separate. Esophagus. The esophageal region of the foregut is narrower than the pharynx. The esophagus has a round lumen and thick walls. A diverticulum near the caudal end of the esophagus at day 6 marks the formation of the crop. The lumen of the esophagus temporarily narrows by days 5 to 6 as the esophageal epithelial lining grows, but opens again by day 8, Figs 11.50b, 11.51b. Stomach. Caudally, the esophagus dilates to form the stomach by the end of day 3 and is visible in 4-day transverse sections, Fig 11.45k. While the stomach migrates to the left of the midline, it divides into the glandular proventriculum and muscular gizzard, Figs 11.51d, 11.53a, 11.55. Duodenum and intestines. The duodenum is visible in transverse sections as a caudal region of the foregut that appears as a vertical oval. It opens to the yolk via the cranial intestinal portal, Figs 11.25p, 11.380, 11.45p. The region of the digestive tract between the stomach and hindgut becomes the small intestine. The ileum of the small intestine becomes convoluted and temporarily herniates, protrudes, into the yolk sac as the intestinal loop. The gut withdraws from the yolk sac during days 17 to 19. Caudal to the small intestine is the large intestine that opens into the cloaca. The caudal 2/3 of the hindgut become the large intestine and cloaca. Specific divisions of the intestine are visible around day 6. Liver and gallbladder. A ventral diverticulum of the floor of the duodenum, called the hepatic diverticulum, gives rise to the liver. The hepatic diverticulum expands into the surrounding mesenchyme that induces the endoderm to proliferate, branch, and differentiate into hepatocytes, liver cells. The hepatic diverticulum branches in two to form the 1. Cranial liver diverticulum, dorsal, that extends toward the embryo's right side, away from the yolk sac, along the ductus venosus, a blood vessel that will be described later, and 2. Caudal liver diverticulum, ventral, lies ventral to the ductus venosus. As it grows, the liver acquires a rich blood supply from the vitelline veins and forms the hepatic sinusoids. Although the liver rudiment begins to form at the end of day 2, it is easier to identify. In 72 and 96-hour embryos, Figs 1138m, L 1.45 kl. The gallbladder and its duct, common bile duct, form as a diverticulum called the ductus jalcdocus from the caudal liver diverticulum. They are visible in transverse sections of 96-hour embryos, Fig LL45L. Whereas the gallbladder develops in chickens, it does not form in all species of birds. Pancreas. Development of the pancreas is closely associated with liver development. The pancreas is derived from 3 rudiments. The dorsal pancreatic rudiment is a dorsal evagination of the duodenum, while the 2 ventral pancreatic rudiments arise as evaginations from the hepatic diverticulum. The pancreatic rudiments are visible on day 4, Fig 11.45m. During day 6, the pancreatic rudiments fuse to form one pancreas with 3 ducts that empty into the duodenum. The adult pancreas produces digestive enzymes. Cloaca and vent. The cloaca is a dilation of the caudal region of the hindgut. It is first visible in transverse sections of 72-hour chick embryos, Fig 11.38v. The vent, anal, region is formed by a caudal ectodermal invagination called the proctodeum, which meets the caudal gut at the cloaca. The resulting membrane of ectoderm and endoderm is called the ducal membrane, Fig 11.38w. At first the cloaca is not open to the outside, but will open when the cloaca membrane ruptures just before hatching. The proctodeum gives rise to the vent. Respiratory system. The respiratory system develops as a diverticulum called the laryngotracheal groove from the floor of the pharynx caudal to the 4th pharyngeal pouches. This is visible as a narrow slit off the pharynx in transverse sections of 72-hour embryos, Fig 11.38h. By 96 hours, the laryngotracheal groove has elongated further, Fig 11.451. The laryngotracheal groove grows caudally and becomes separated from the digestive system as a tube called the trachea. The cranial end of the laryngotracheal groove remains open to the pharynx as the glottis. A pair of lateral diverticula called lung buds evaginate from the caudal end of the laryngotracheal groove to produce the future bronchi and lungs. Lung buds grow into the surrounding mesenchyme and appear as small donuts ventral to the esophagus in transverse sections at 3 to 5 days, Figs 1L.38ij. 11.451j, 11.48. The epithelial lining of the respiratory system is derived from pharyngeal endoderm, and the surrounding mesenchyme forms muscles, connective tissue, and blood vessels. By day 6, the cranial end of the trachea expands to form the larynx that lacks vocal cords in birds. The syrinx, which is the avian vocal organ, forms at the junction between the trachea and bronchi, mesobronchi. In the adult, air is taken into the respiratory system through the external nares and enters the mouth through the internal nares, passes into the glottis, larynx, trachea, and bronchi before entering the lungs and air sacs. The lungs are tearized by numerous parabronchi that permit one-way airflow instead of blind-ending alveoli with two-way airflow in mammals. The lungs appear sponge-like in 8 and 12-day embryos, Figs 11.51, 11.55. The lungs are located in the pleural cavities that are divisions of the intraembryonic xylem. The avian respiratory system is also characterized by 9 air sacs that are outgrowths from the lungs that remain connected to the lungs. The air sacs are found among the viscera and in some of the long bones, Fig 11.51. Skeletal and muscular systems. The skeletal system is derived from neural crest, mesenchyme, and paraxial mesoderm. Muscles are derived from 3 sources: 1. Mesenchyme, which generates smooth muscles of blood vessels and some viscera; 2. Splanchnic mesoderm, which produces smooth muscle of the gut and its derivatives, and the muscular heart; and 3. Paraxial mesoid cream, that produces voluntary muscles. As described in Chapter 9, the segmental plate of the paraxial mesoderm gives rise to paired somates during somatogenesis that form by 24 hours, Fig 8.371. The somates generate cells that produce cartilage of vertebrae and ribs, muscles of the rib cage, limbs, abdominal wall, back and tongue, and dermis of the dorsal skin. By 48 hours, the somates have differentiated into 3 regions from which these structures are derived: chromatome, dermis of dorsal skin; myotome, muscles; and sclerotome, vertebral skeleton, Figs 9.4b, 11.25. Somates are formed and differentiate in a cranial to caudal direction. All 50 of the somite pairs develop by 4 days. Dermarome cells form the outer motif part of the somite. They appear dark in transverse sections and are located lateral to the myotome. Myotome cells appear as a long light stripe lateral to the sclerotome, and extend toward the caudal cardinal vein. At 2 to 5 days, the dark sclerotome cells appear loosely arranged lateral to the spinal cord and notochord. Sclerotome cells migrate around the spinal cord and notochord to form the 40 vertebrae of the adult chicken: 14 cervical, 7 thoracic, 5 lumbar, 2 sacral, and 10 caudal, and pygostyle, several fused vertebrae. Avian vertebrae are composed of a vertebral body, centrum, ventral to the spinal cord, and neural arches dorsal and lateral to the spinal cord, Figs 11.51c, 11.53b. The medial region of the sclerotome forms the vertebral body. The lateral part of the caudal region of the somite contributes to the neural arches and pedicles of vertebrae and to the ribs. By day 5, the vertebrae are cartilaginous and begin ossification, bone formation, around day 13. The somatic mesoderm of the lateral plate mesoderm gives rise to the sternum, ventral muscles, pectoral girdle, scapula, coracoid, clavicle, and the pelvic girdle, ilium, ischium, pubis, Figs 1.53, 11.55. The somatic mesoderm generates the skeleton of the limbs. Mesenchyme of the paired wing buds gives rise to the bones of the wing, humerus, radius, ulna, carpals, metacarpals, phalanges, after day 4. Somatic mesoderm also generates the bones of the hind limb, femur, tibia, fibula, tibiotarsus, tarso-metatarsus, phalanges. Myotome cells contribute to limb muscles. The apical ectodermal ridge is a thickened ectoderm structure located at the tip of each limb bud that is an important signaling center for the developing limb buds. Without it, the limb fails to grow. The apical ectodermal ridge is first visible at the tip of 72-hour limb buds, Fig. 1L.38 pu. Cranial neural crest cells form the pharyngeal arch and craniofacial bones and cartilage. Bones of the skull are thought to arise from the neural crest, paraxial mesoderm, and the first 5 somates. The skull is formed from membranous bones and cartilage. The cartilage begins to form by day 5 and the bone begins ossification on day 9. Each scleral papilla that is located around the margin of the cornea of the eye induces a scleral ossicle, small eye bone, to form by day 10, Fig 11.52. Whereas the myotomes generate the voluntary muscles of the dorsal and dorsolateral regions of the trunk and limbs, the somatic mesoderm produces the muscles of the lateral and ventral body wall, and tendons of the limbs. The muscles of the head are derived from cranial neural crest, head mesenchyme, somates, and somatic mesoderm. Tongue muscles are derived from myotomes of somates. 1 to 5, and mesoderm of the first 3 pharyngeal arches. Most of the body muscles appear around days 5 to 9 and take on the adult arrangement by day 10. Although muscle identification of the embryo is beyond the scope of this atlas, muscles can be observed in sections of later stage embryos, Figs 11.51, 11.53, 11.55. Splanchnic mesoderm generates the smooth muscles of the alimentary tract and its associated structures. Splanchnic mesoderm also generates the heart, to be discussed later. Urogenital system. The urinary system and the genital, reproductive, system will be considered together because they originate from similar tissues and share many of the same ducts as they develop. The urinary system functions to eliminate wastes and consists of the paired kidneys and ducts that transport urine to the cloaca. The genital system functions in reproduction and includes the gonads and ducts that are used to transport gametes to the cloaca. The intermediate mesoderm gives rise to most of the urogenital system. The cloaca is derived from endoderm and the reproductive cells develop from primordial germ cells, Fig 9.14. Urinary system kidney development occurs in 3 stages in which 3 different, paired excretory organs form in the embryo, pronephros, mesonephros, and metanephros. They will be discussed as though a single organ here. They develop from cranial to caudal. The pronephros develops at the level of somates 5 to 15, mesonephros at somite levels 13 to 30, and metanephros at somite levels 31 to 33. The pronephros develops first, by 36 hours, and is the most cranial. It is rudimentary and non-functional in the avian embryo. Small vestiges of the pronephros may be observed in transverse sections of 72-hour chick embryos as tiny tubes or darkened areas ventrolateral to the dorsal aorta at a level rostral to the cranial intestinal portal. It begins to degenerate by about 43 hours stage 1-3, and is absent by day 8. The mesonephros is the functioning kidney of the embryo. It extracts nitrogenous wastes from the blood and produces urea at first and then uric acid. The mesonephros forms on the mesonephric ridge. It is a rounded bulge that forms on the dorsal body wall and extends into the peritoneal cavity. The mesonephros consists of a single mesonephric duct and many mesonephric tubules that are S-shaped and extend medially from the mesonephric ducts. Each mesonephric tubule produces a cup-shaped end, glomerular capsule, that surrounds a tuft of capillaries, glomerulus. The mesonephric duct is visible ventral to the caudal cardinal vein as the lateral area of the mesonephros in transverse sections of 48 to 96 hour.
Embryos, Figs 1125, 1138 QT, 1145 PW. In 96 hour embryos, the mesonephric duct as dilated and appears more prominent before joining the cloaca. The mesonephros functions by day 5 until about day 11 when it degenerates and the metanephros functions, Figs 11.51d.
1.53. In males, the mesonephric duct persists as the nephric duct, Wolffian duct, which later becomes the vas deferens. Wastes filtered by the glomerular capsule enter the mesonephric tubules, and flow into the mesonephric ducts before entering the cloaca and passing in the allantois where they are stored. The mesonephros is the functioning permanent kidney and is the only kidney found in the adult. It develops as a diverticulum, ureteric diverticulum, metanephric diverticulum, at the base of the nephric duct at the end of the day 4. The ureteric diverticulum grows dorsally into the mesonephric ridge and expands to form the metanephros. The narrow region is the metanephric duct, which is the future ureter. In transverse sections, the metanephros appears lateral to the dorsal aorta and medial to the mesonephros, Figs 1.51, 1.53. The metanephros begins excreting by day 1-1. The adult chicken metanephros has 3 elongated lobes rather than the typical mammalian bean shape. Reproductive system. The gonads develop on the genital ridge, which is a swelling of the splanchnic mesoderm located on the dorsal wall of the chylome. The primordial germ cells migrate from the germinal crescent, anterior region of the area pellucida, through the bloodstream T0 the peritoneal epithelium and colonize the gonad. Sex cords of the testis arise about days 5 to 6. By day 13, the germ cells have differentiated into spermatogonia. Although primordial germ cells migrate to both ovaries, the right ovary is rudimentary in the chicken while the left ovary becomes the functioning ovary of the adult. Oogonia are present during day 7. By day 8, sections of the gonad reveal enlarged primary sex cords characteristic of the ovary. By day 14, macroscopic examination of the embryo reveals its gender. The oviducts, Mullerian ducts, develop adjacent to the nephric duct, Fig 11.51d. The right Mullerian duct in female chicks and both Mullerian ducts in males begin to degenerate on day 8, and are gone by day 12 in males and day 18 in females. In females, the left Mullerian duct fuses with the cloaca about day 11. Circulatory system. General highlights of cardiogenesis and vasculogenesis were covered in Chapter 9, and understanding these processes is critical to comprehend embryonic circulation. Specific structures of interest will be described below. For simplicity, paired structures are indicated by an asterisk, and are described as though they were single structures. When embryos are injected with black ink, the path of the blood through the heart and blood vessels is readily apparent, Figs 11.15, 11.21, 11.27, 11.34, 11.41. Heart. The heart arises from two areas of splanchnic mesoderm, each of which forms an endocardial tube that fuses in the midline to form a single heart tube in the neck region. Thickened splanchnic mesoderm is first visible in the 24-hour chick embryo, Figs 8.35, 8.37g. This forms the proximal wall of a large amniocardiac vesicle, which is the cardiac portion of the chylome that develops into the pericardia cavity. The fusing endocardial tubes can be seen in transverse sections of 28-hour embryos, Fig 11.4e. By 33 hours, the heart lies cranial to the cranial intestinal portal and is bent to the right, Figs 11.5 to 10. At this stage the walls of the heart have differentiated into, 1. The thin, inner endocardium which forms an inner tube that will later become the epithelial lining of the heart, and 2. The outer, thicker myocardium that will differentiate into the epithelial covering of the heart, epicardium, and muscular wall of the heart, myocardium. At 33 hours, the heart, which is somewhat U-shaped, has three regions listed from cranial to caudal: 1. Cona truncus, bulbus cordis, conus arteriosus, bulbus arteriosus, which is the cranial narrower arm of the U that gives rise to the future truncu, arteriosus, and aortic sac that generates the ventral aorta; 2. Ventricle, which forms the base of the U; and 3. Sinoatrial region, which is the caudal portion of the U that is immediately cranial to the cranial intestinal portal, Figs 11.6, 11.7, 1. 1.10, 11.13, 11. 15. The sinoatrial region will differentiate into a cranial atrium and caudal inu venosus by 40 hours. Figure 11.16. Note that in the 33-hour embryo, the sinoatrial region bifurcates into the Vauquelin veins, omphallomesenteric veins, that empty into the sinus venosus. The heart loops, bends, to the embryo's right by 40 hours, Fig 1.16. At 43 hours, the heart is coiled on itself and has expanded. Figs 11.17, 11.18c. Focusing up and down on the heart of 48-hour whole mounts is critical to observe the following areas: cona truncus, visible at high focus lying parallel or at an oblique angle to the hindbrain; ventricle at high focus appears as a large vesicle continuous with the cona truncus and bends at a right angle toward the embryo; atrium, at low focus, is a curved region of the heart that arches under the cona truncus; and sinus venosus, at low focus, appears as a small chamber caudal to the atrium. Figs 11.19 to 21. The relationships of these structures can be observed more clearly in sagittal and transverse sections of 48-hour embryos, Figs 11.23, 11.25. Both the conotruncus and ventricle have a recognizable endocardium and myocardium identifiable in 48-hour transverse sections, Fig. 1.25. At 72 hours, the heart is more coiled and appears five-shaped since the ventricle has looped or bent to the embryo's right and the atrium begins to travel above the ventricle. The heart has also enlarged, making the regions of the heart easier to distinguish in whole mounts and sections, Figs. 1.29-33, 11.36, 11.38. The atria have extended laterally. The wall of the ventricle is thickened in transverse sections of 72 and 96-hour embryos, Figs. 11.38, 11.45. The incratrial septum divides the atrium into two separate compartments. It forms by day 4, and is more prominent in day 5 and older embryos, Figs. 1.45, 11.48. The incratricular divides the ventricle into two separate compartments by day 7 to 8, Fig. 11.51. Invasion of the cardiac neural crest cells into the conotruncus truncus arteriosus begins to separate the conotruncus into the pulmonary artery and aorta on day 5, Fig. 11.48. The heart changes its location from the neck area at the level of the rhombencephalon when it first forms, to the level of the wing buds, Fig. 11.51d. The heart first begins to beat weakly when the embryo has developed 9 somates at about 28 hours. The right side of the adult heart receives deoxygenated blood from the body's tissues and pumps it to the pulmonary system of vessels. The left side receives oxygenated blood that it distributes to the rest of the body. In summary, the adult avian heart has 4 chambers that arise from 2 chambers, an atrium and a ventricle that arise from 2 endocardial tubes derived from splanchnic mesoderm. Embryonic circulation. There are 3 major routes of embryonic circulation or circulatory arcs in the chick embryo, with the heart as the pump at the center of each. The Viklin circulation carries blood to and from the yolk sac for nutrition. 2. The allantoic circulation carries blood to and from the area just beneath the eggshell where gas exchange occurs. These 2 circulatory arcs are considered extraembryonic circulation because they lie mostly outside the embryo. 3. The intraembryonic circulation is confined to the body of the embryo and functions to bring nutrients and oxygen that were collected by the other circulatory arcs to the embryo, and carry wastes from the embryo to either the allantoic circulation or the mesonephros when it becomes functional. Extraembryonic blood vessels. The extraembryonic blood vessels are produced in the extraembryonic membranes of the area vasculosa. The Viklin blood vessels are derived from blood islands in the splanchnic mesoderm of the area opaca. The vitellin blood vessels carry blood from the embryo to the yolk sac and back again. Circulating blood leaves the heart and passes through the ventral aorta, through the aortic arches and dorsal aorta out through the left and right vitellin arteries, omphallomesenteric arteries, to the plexus of blood vessels on the yolk. After absorbing nutrients from the yolk and oxygen, which passed through the eggshell and albumin, the blood collects in the circular sinus criminalis and drains into the cranial and caudal vitelline veins, which merge into a single large vessel, the ductus venosus, before returning to the sinus venosus of the heart. The extraembryonic blood vessels are easily recognized in embryos injected with black ink, Figs. 11.18, 11.21, 11.27, 11.34, 11.41. By day 4, the allantois has begun development and its allantoic blood vessels rake over the role of oxygenating the blood and exchanging gases, Figs. 11.1, 11.41. Intraembryon C blood vessels. Blood that is destined for circulation within the embryo leaves the heart and passes through the ventral aorta, through the aortic arches, and into the dorsal aorta, where it is distributed to the body. The cranial cardinal veins return blood from the cephalic region of the body, and the caudal cardinal veins drain blood from the caudal part of the body. Near the heart, the cranial and caudal cardinal veins fuse together to form a single common cardinal vein, which drains into the sinus venosus. Blood from the intraembryonic and extraembryonic circulation mixes in the heart, allowing exchange of nutrients, wastes, and gases. Specific intraembryonic blood vessels are described below. Dorsat A or La. The dorsal aortae are first visible ventral to the somates and lateral to the notochord in transverse sections of 28-hour embryos, Fig 11.4 gh. The dorsal aortae are connected to the short ventral aorta, which runs from the cona truncus to the pharynx floor, via the aortic arches that are first visible in transverse sections at 33 hours, Fig 1.1.15. Cranial extensions of the dorsal aorta become the internal carotid arteries, which are identifiable in transverse sections of 72-hour embryos, Fig 11.38 gk, and become enlarged by 5 to 6 days, Figs 11.47, 11.48, 11.50. Caudally, the dorsal aortae fuse to form a single, median descendant aorta that more caudally splits again into two dorsal aortae, Figs 1.21, 11.27. By 96 hours, the dorsal aorta extends into the tail as the caudal artery, Figs 11.41, 11.45. Branches of the dorsal aorta include the intersegmental arteries, segmental arteries, which supply the dorsal organs such as the somates, neural tube derivatives and notochord, Figs 11.27, 11.34, 11.41, 11.45. The descending aorta gives rise to arteries supplying the limbs, vitelline arteries, mesonephric arteries, and allantoic arteries, umbilical arteries. Ventral aorta. The ventral aorta extends cranially from the aortic sac, an extension of the cona truncus, and gives rise to the aortic arches. The ventral aorta bifurcates and extends cranially to form the external carotid arteries that are visible by 96 hours, Fig 11.41. By day 7, the internal and external carotid arteries anastomose, fuse together. The carotid arteries branch to supply the arteries of the head. Aortic arches. The paired aortic arches supply the paired pharyngeal arches, and are a communication between the dorsal and ventral aorta. They do not form simultaneously, but instead develop in a cranial to caudal direction. The aortic arches encircle the pharynx. The aortic arches are visible in the corresponding pharyngeal arches of embryos from 48 to 96 hours. The first aortic arch is visible at 43 hours, Fig 1118, the second and third aortic arches by 48 hours, Figs 11.21, 11.25, the fourth aortic arch by 72 hours, Fig 11.38, and the sixth aortic arch by 96 hours, Figs 11.41, 11.45. The fifth aortic arch is transitory and disappears soon after it forms. Modifications of the aortic arches to the adult form are shown in Fig 9.17. Venous system. When viewed from the dorsal side, the venous system in the 3-day chick embryo appears shaped like the letter H. The paired cranial and caudal cardinal veins form the vertical portion of the H, and the horizontal portion of the H is formed by the common cardinal veins which bring blood to the sinus venosus, at the center of the horizontal bar of the H. The cranial cardinal veins are first recognized in transverse sections of 33-hour embryos, Figs 11.15. By 43 hours, they are large veins visible in the cephalic region of the embryo, and they remain large during each stage of development shown in this atlas, Fig 11.18. They give rise to the internal jugular veins and ventral veins of the neck. Each cranial cardinal vein drains into the common cardinal vein * visible at 48 hours in transverse sections, Fig 11.25n. Cranially, the caudal cardinal vein also enters the common cardinal vein. The caudal cardinal vein is readily identified as lying lateral to the dorsal aorta, caudal to the level of the common cardinal vein in injected hole mounts or transverse sections of 48-hour or older embryos, Figs 11.25, 11.34, 11.38. When the mesonephros is present, the caudal cardinal vein lies immediately dorsal to it, Figs 11.25, 11.38. Another vein that drains into the common cardinal vein is the allantoic vein, umbilical vein. That is visible in the lateral body wall in transverse sections of 72-hour or older embryos, Figs 11.38, 11.45. The merged vitelline veins form the ductus venosus that enters the sinus venous of the heart. The liver forms around the ductus venosus, Fig 11.45. By day 5 the vitelline vein has differentiated into the hepatic portal vein. INT egument. The INT egument is composed of the epidermis on the body surface, and the dermis lying immediately under the epidermis. Between the INT egument and deep body musculature is the hypodermis, which is loose connective tissue and adipose tissue. The epidermis is derived from ectoderm, and the dermis is derived from mesoderm, dermatome, somatic mesoderm, and neural crest. Initially the ectoderm is a single layer, but forms an outer, temporary protective layer called the periderm by about 43 hours that is sloughed off. By day 18. Our discussion of the INT egument will also include differentiated structures of the skin, feathers, beak, comb, wattles, scales, and claws. Feathers form in distinct tracts, pterally, on the body surface, Fig 11.58. The most common feather of adult birds is the contour feather, which has a long shaft from which many barbs arise along its length. IR functions to give the bird's surface an aerodynamic shape. The first feathers to form in the embryo are down feather, also called plumules, Fig 11.57. They have a short shaft from which the barbs arise in a circle at the same level. Down feathers stay close to the skin and are for thermal insulation. Feathers develop from epidermal placodes that arise from epithelial mesenchymal interactions. Under each epidermal placode the dermal cells condense to form a dermal papilla. The epidermal placodes form invaginations of the epidermis into the underlying dermis as they give rise to feather papillae, feather buds, that develop into feather follicles by day 20 and feathers, Figs 11.58 to 60. Feather rudiments appear as white dots at about 5 days, first on the lumbar surface, and later on other surfaces. Beak, COJ/IB, and wattles in chickens, the jaws are drawn out into a beak, Fig 118. The beak begins to form from the upper and lower jaws by day 6. On day 7, an egg tooth forms on the tip of the dorsal side of the upper jaw, Figs 11.54, 11.56, 1158. The egg tooth is thought to aid in breaking open the shell during hatching. The beak becomes keratinized, and by day 14 the jaw is hardened. The comb develops on top of the head as a thickened, medial dermal ridge beneath the epidermis beginning on days 6 to 7. It is serrated by day 12, Figs 11.55, 11.56. The wattles are fleshy, pendulous epidermal structures with a mesoderm core. The wattles hang from the lateral sides of the lower beak. The wattles begin to appear by day 11, developing similar to the comb, Fig 1.56a. Scales and claws. The epidermis of the avian leg and foot is covered with scales that arise as a series of placodes on about day 10 that elongate distally until day 17. The scales overlap and become keratinized, Fig 11.56. Scales are formed as a result of complex epithelial mesenchymal interactions, and thus scale formation is reminiscent of feather development. An exception is the lack of dermal papillae. Claws are modified scales that form at the end of the toes by day 10, Figs 11.54, 11.56. Abnormal development. Common type of anomaly, abnormality, is duplication of the primitive streak. This results in conjoined twins. Figs 11.64 to 11.67. Other abnormalities include microphthalmia, small eyes, cyclopia, one central eye, and stunted embryos. It is unclear what causes each anomaly since there can be both genetic and environmental causes. Some anomalies, example primitive streak duplication, appear to be due to incubation of aged eggs or brief incubation at inappropriate temperatures.